this liver seminars edition. I am Ana Blas Garcia from the University of Valencia, and it's a great honor for me to participate in this seminar series. I would like to thank the organizers, Dr. Rafael Bañares and Dr. Jordi Gracia Sancho, for giving me the opportunity to host this young investigator seminar, which is entitled Lipotoxicity in Adipose Tissue Macrophage as Link Between Obesity and Its Complications. It is my pleasure to introduce you to today's speaker, Dr. Guillaume Bidault from the University of Cambridge. Dr. Bidault obtained his PhD from Sorbonne University in Paris in 2013, working on the molecular mechanisms leading to lipodystrophic and premature aging syndrome due to mutations in the lamin gene. Since late 2013, he is a postdoctoral researcher at the University of Cambridge in the group of Dr. Vidal-Puig, where he has mainly studied how the infiltrations of macrophages in the adipose tissue contributes to the metabolic alterations of obesity. Currently, he investigates the role of lipid metabolism and lipotoxicity in macrophages as a link between obesity and its cardiometabolic complications, such as fatty liver disease, diabetes, and cardiovascular diseases. Before starting with Dr. Bidault's presentation, I would like to remind you that we will have time for discussion after his talk. So please do not hesitate to write your questions or your comments down on the tab, or to raise your hand later to ask by yourselves. So thank you very much, Guillaume, for accepting our invitation and for being with us today. And whenever you are ready.
Yeah, so thanks a lot, Hannah, for these very, very kind introductions, and also to the organizer and yourself for providing me the opportunity to present today on this liver seminar. I'd like also to thank Anna's students, Isabel, who've been doing some times in our lab last year, and she thought it was that those seminars were a good fit for having me presenting and suggested to Hannah. So today I'm going to speak to you about the role of lipotoxicity in adipose tissue macrophages as a potential link between obesity and its complications. As you know, obese people have a susceptibility to develop a lot of disease. People are obese for various reasons. The main one is being on a positive energy balance, which is the result of an excess caloric intake, bad feeding behavior, a sedentary lifestyle, which reduces the number of calories that are burnt every day. Also some environmental effect and some genetics. We are obviously not all equal when facing metabolic diseases. So when on a positive energy balance for a certain period of times, people tend to gain weight, and this weight gain is associated with the development of complications, as I was mentioning, such as insulin resistance, dyslipidemia, hypertension, diabetes, NAFLD, and I will come to that a bit more in detail later, and cardiovascular disease or cancer. Quite interestingly, the development of NAFLD in obese patients is one of the most common comorbidity associated with obesity, and the last figures released few months ago, were showing that around 75% of people with obesity will develop NAFLD, have NAFLD, and 34% of them will develop the inflammatory form of the NAFLD NASH, which is a major problem for our society. And I think this audience knows more than I do about the deleterious consequences of NASH in the population. However, having a bit of extra kilos doesn't necessarily make you having NAFLD. And there is actually a bit of a paradox, which is the lean NAFLD paradox, and which is notably present in some of the countries in Asia. And those people tend to develop fatty liver disease without being obese. As you can see here, that's from 5 to 20%, depending on the studies, of people with NAFLD are actually lean people. And this could be even more pronounced if you look to a very, very peculiar population, which is the people with lipodystrophic syndromes due to either genetic alterations or even acquired form that do not have very few fats or fat in the wrong place. And this is highlighted here by this mouse model, which is the AZIP mouse described a long time ago, actually. And this mice, as you can see here, do not have any fat deposit. They don't have any adiposity, despite though they have very, very strong fatty liver disease development, such showing somehow that you don't necessarily have to be obese to develop NAFLD. And to explain this apparent paradox, our laboratory have been developing the adipose tissue expandability hypothesis. And basically, the adipose tissue expandability hypothesis suggests that every single individual has their own capacity to store the excess energy when on a positive energy balance in the form of lipids within the adipose tissue, and more specifically within the adipocyte cells, which are the cells that are professional to store lipids in the body, as you're certainly aware. However, in various situations, such as obesity, aging, or lipodystrophic syndromes, as example, this capacity to store excess energy might be exceeded. And when this is happening, this excess energy in form of fat do not store anymore within the adipose tissue, but will be stored somewhere else, in other organs and cells. This is happening and has been described to happen in the muscles, in the heart, in the artery, or in the liver. And this ectopic lipid accumulation is known to participate to the development of the cardiometabolic complication of obesity, such as insulin resistance, fatty liver disease, NAFLD and NASH, atherosclerosis, or heart failure. We and others actually observed that some of the cells composing the adipose tissue, non-adipocyte cells, do accumulate as well lipids during obesity. And we have been observing that when lipid accumulation occurs within adipose tissue macrophages, those macrophages tend to be activated. They tend to be inflamed. And these activations of the adipose tissue macrophages has two effects. First of all, it renders even sicker the adipose tissue cells, impairing even more the capacity of this tissue to store excess energy inside. And it's also trigger inflammations that is known to participate to the development of the cardiometabolic complication of obesity, notably fatty liver diseases. However, recent evidence has been showing that having fats inside your macrophage is not necessarily a bad thing. And in 2019, this paper has been published in Cells showing that very specific populations of macrophages within the adipose tissue will be full of fat, will express a series of markers such as CD9 or TREM2, and they're actually scavenger cells that are found in the chrome-like structures surrounding the dying adipocyte. And if you can't make those cells by deleting notably the molecule TREM2, your adipose tissue will become even sicker. You will have an adipocyte hypertrophy, your level of insulin will be higher, your blood cholesterol will be higher, you have increased glucose intolerance. So those lipid-associated macrophages, at least in some context, seems to be protective against the effect of obesity in adipose tissue, and therefore later on in the whole organism. However, we did observe in our data sets that the picture is not as simple as those macrophages being simply protective. So in the laboratory, we have data sets that is adipose tissue macrophages isolated from different stages of the disease. So we use the OB-OB mouse, the hyperphagic OB-OB models to remove any dietary effect because they eat the same food as their wild-type litter mates. And we took those animals at two different ages. The first one is at 5 weeks of age when the mouse is already obese, but the tissue is still expanding. It's sort of a healthy, metabolically healthy obesity state. And we also took these mice at 16 weeks of age when the adipose tissue capacity to store is overwhelmed. Those mice are frankly insulin resistant. They are very, very strong NAFLD. And we isolated the macrophages from those two animals. And quite interestingly, if you look to the expression within those macrophages of the markers of lipid-associated macrophages, you can see that they are equally upregulated when comparing to the wild-type glutamate, either when the tissue is still healthy or when the tissue is not healthy anymore, it's metabolically impaired. And indeed, at 5 weeks of age in our OB mouse, you can see that those macrophages do express a lot of the anti-inflammatory M2-like markers, not much of the pro-inflammatory cytokines such as TNF-alpha, while later on in the life, when those mice become really, really sick, you lose the anti-inflammatory profile of those cells, and you gain a pro-inflammatory profile with an increased expression of MCP1, TNF-alpha, CD11C, or interferon beta, as we published many, many years ago. So actually, it seems that you can have those lipid-associated macrophages, but they are not always beneficial. Sometimes they seem to be good and anti-inflammatory, and sometimes they're not. And looking a bit to what happens to the liver, despite it's not my main field, I was seeing that the very, very same thing seems to happen in NAFLD and NASH. In that publications published last year, the author of this study has been demonstrating that some macrophages within the liver accumulate fat in a macrophage scavenger receptor 1 fashion. And if you delete MSR1 within those macrophages, the liver becomes less inflamed and less fibrotic. So it seems in that context that those foamy macrophages are deleterious. Similarly, 2 years ago, this paper has been published that there is two apparently Kupffer cell populations within the liver, KC1 and KC2, and the KC2 populations express the lipid transporter CD36. And when you remove CD36 from those macrophages, or you ablate those Kupffer cell type 2, the mice will not develop NAFLD in the context of a high-fat diet feeding. So showing that it seems that those foamy macrophages in NAFLD or NASH could be deleterious. However, last year and 2 years ago, among other papers, it's been shown that some lipid-associated macrophages, LAMs, here as you can see, can develop from monocyte-derived macrophages in the liver in NASH. And actually, those lipid-laden macrophages are anti-inflammatory, form clone-like structures surrounding lipid-laden apoptotic hepatocyte, and that is reducing fibrosis. Quite interestingly, it's been shown last year that this system of scavenging of efferocytosis of those lipid-laden apoptotic hepatocytes tends to fail due to the loss of TREM2 protein at the surface of the macrophages, and this further develops the inflammations of the liver and promotes NASH progression. So just to summarize, and coming back a bit more to adipose tissue, in a lean or a metabolically healthy obesity state, we know that lipids can travel from adipocyte to macrophages, and that actually it seems that macrophages are perfectly able to deal with them. They accommodate with those lipid fluxes. And it seems that actually this is important, and that macrophages are able to signal and maintain tissue homeostasis. However, in obesity, in metabolically unhealthy obesity, when the capacity of those adipocytes to store properly and to be healthy storing fat has been exceeded, a lot of lipids will traveling into the body and notably inside the adipose tissue macrophages. And your cells might retain the capacity to deal with this excess fat, but having too much of it or a bad quality of it or being in the wrong environment trigger ER stress, metabolic inflammation, and cell death. And we know that this participates to adipose tissue dysfunctions, liver diseases, cardiometabolic diseases associated with obesity. So the idea we have in the lab is that what if those macrophages could deal better with lipids? Can we reduce metabolic inflammation and therefore reduce the cardiometabolic complication associated with obesity? However, I show you different paradoxes so far in which we have lipid accumulations in macrophages, in the adipose tissue, and in the liver. But sometimes the macrophages get sick of it, and sometimes they don't. And why is that is actually largely unknown. What we know from epidemiological studies is that the quality of the lipids matters. And there's been several studies, and I'm just highlighting two over there, showing that in particular, saturated fatty acid intake, notably and mostly in the form of palmitic acids, the C16O lipid, is associated with an increased risk of coronary heart disease, independently of any coronary heart disease risk factor. The same is true when you look at the saturated fatty acid serum level that are predictors of the occurrence of type 2 diabetes mellitus. And as you can see here, even still, when you adjust for the classical risk factors of type 2 diabetes mellitus, such as age, sex, BMI, waist ratio, etc. So it seems that the quality of the fats is important, and that saturated fatty acids are the one linking obesity and dyslipidemia to its complications. And at the level of inflammations, it's been long known that palmitate and saturated fatty acid are pro-inflammatory. And one of the views that was prevailing in the field for many, many years is that that palmitic acids, saturated fatty acid, were agonists for the tool-like receptor 4, which is the receptor for the lipopolysaccharide, which is a component of the gram-negative bacteria. However, this view has been strongly challenged over the years, and I think this paper definitely ends this view, showing that TLR4 is not a receptor for saturated fatty acid. However, inflamed macrophages have a reprogrammed lipid metabolism, rendering palmitic acid inside the cells inflammatory. So the message I want to say is that actually here, it seems that palmitate do not signal to receptors, but need to be inside the cells to exert its inflammatory action. And going to a very simplistic view, the way you can get saturated fatty acids within your cell is quite simple. There are two routes. Either you take it up or you make it up through the de novo lipogenesis pathway. And on my talk today, I'm going to present to you results about both aspects of how cells make or uptake and deal with saturated fatty acids. But starting with the de novo lipogenesis pathway and trying to see how it's regulated de novo lipogenesis in obese adipose tissue macrophages. So we took our beloved data set of healthy and unhealthy adipose tissue macrophages from obese, and we look into the expressions of the de novo lipogenesis pathway, which you know is under the transcriptional regulations of SREBP1. And what we did observe is that in these mouse that are still healthy, obese, and healthy, the target genes of SREBP1 are upregulated, while they get downregulated when the mouse is sick. And this is actually quite correlated with an increase of the alternative activation profile of the macrophages at this age, since some adaptive mechanisms in the development of obesity that is lost basically in obese, morbidly obese animals. So we were wondering if this SREBP1 activation that we observe concomitantly to the increase of the alternative activations, that the increase of SREBP1 activations that is concomitant to the increase of the M2 profile of the adipose tissue macrophages, we were wondering if SREB1 activation is actually a feature of those M2 kind of macrophages. So to do that, we used first a publicly available dataset from human macrophages that were either treated with interleukin-4, which is the canonical cytokines to trigger an alternative activation of the macrophages, or with LPS, lipopolysaccharides, which is a typical trigger of the classic activation of the macrophages. And when we perform a transcriptional regulator prediction analysis, What we observed, first of all, is that our analysis work. Indeed, NF-κB is upregulated in response to LPS. STAT6, classical transcription factor for alternative activation, is upregulated. We observed that actually SRABP1 is predicted to be very, very, very strongly activated in human macrophages following interleukin-4 activation when compared to LPS, to classically activated macrophages. We then perform RNA sequencing analysis in urine, bone marrow-derived macrophages. Over there, performed a similar analysis. And once again, we found that SRABP1, amongst already described important transcription factors for alternative activation, such as STAT6 or PPAR gamma, was predicted to be strongly upregulated. We then confirmed those findings in vivo by injecting C57 Black 6's mouse with interleukin-4 and sacrificing them 2 days after. We collected the peritoneal macrophages from those animals and ran a few genes from those cells, and we did observe that indeed SRABP1 is activated in vivo macrophages that has been polarized into an alternatively activated state. So we saw that SRABP1 is activated in alternatively activated macrophages, but the next question we wanted to ask was to know whether SRABP1 activation is necessary and relevant for this macrophage alternative activation. So for this, we went quite away from our metabolic remit, trying to find what would be the best model for alternative activation. And the best model for alternative activations is infection with parasitic helminths, basically worms that still infect the vast majority of the population in the world. So we went to some collaborators in the laboratory of molecular biology in Cambridge, and we subjected our mouse that are either wild types or SCAP knockout within the myeloid population. So if you don't have SCAP, as you're probably aware of, you can't activate the SREB transcription factors. So therefore, we have a model here that cannot activate SRABP1 specifically in the myeloid compartment, so basically in the macrophages. So we subjected wild type or SCAP knockout mouse to a worms infection. And these worms will go into the lung, where it will grow. The larvae will go to the lung, where they will develop and then move to the gut to be eventually expelled. So as expected, the inoculations of the larvae of those parasites induce an increased number of macrophages within the lung, as you can see here on the wild type cell. And most of those macrophages are alternatively activated as expected from this challenge. However, what we did observe is that if you cannot activate SRABP1 within your macrophage population, first of all, you have less macrophages, and those macrophages are less able to polarize into an alternatively activated state. And eventually, that leads to a worsened disease progression. Indeed, you can see here that the mouse that could not activate SREB1 in their macrophages have an increased bleeding hemorrhagic lung, as you can see here, that's calculated by the number of red blood cells in the lungs, and they have a delay in the expel of the worms, and we found more of the worms in the gut of the cells. So basically, mice that cannot activate SREB1 in their macrophages have an impaired immune response to parasitic helminths, which involve the alternative activation of the macrophages. So we've been demonstrating that SRABP1 activation is happening in alternatively activated macrophages and is relevant in a disease context. But why SRABP1 activation is important for macrophage alternative activation, this was still unknown. One of the key pathways that came to mind is that those macrophages cannot activate SREB1, they cannot do de novo lipogenesis. So what we did is we used in vitro bone marrow-derived macrophages. We treated them with interleukin-4. They polarized very well into alternatively activated macrophages. And then we use rising doses of the fatty acid synthase inhibitors in C75. And as you can see here, the more you use the inhibitor, the less the cells are able to engage into an alternative activation state. So that was like all looking very, very good until we tried to rescue the phenotype using the end products of the de novo lipogenesis. As you can see here, when we supplemented the cells that have an impaired alternative activation due to the treatment with the fatty acid synthase inhibitor SYN75, we could not rescue the alternative activation impairment due to the blockade of the fatty acid synthesis. So it seems that this is not the lipids that are produced through lipogenesis that are important for the alternative activations. So we ask ourselves the question, which process is regulated by fatty acid synthesis but is independent of the production of lipids themselves? So we've been looking into sequencing data of those macrophages, wild type and SCAP knockout treated with interleukin-4. And what we observed, first of all, is that interleukin-4, and by the time was like complete surprise, and this has been confirmed by several other publications, interleukin-4 seems to induce some reprogramming into the redox homeostasis of the macrophages. As you can see here, pathways associated with redox homeostasis are upregulated in response to interleukin-4 in the wild type cells. However, what we did observe is that the knockout cell, the SCAP knockout cell, failed to upregulate the pathway associated with redox homeostasis in the response to IL-4. So then comes the next obvious question: how come a lipid synthetic program could regulate redox homeostasis? And to make a long story short, and a lot of failed experiments, to be honest, what we discovered was going back to the biochemistry book. Basically, what does fatty acid synthesis that could interfere with the redox status of the cells? And what we know is that fatty acid synthase requires the cofactors NADPH to work. And actually, for each molecule of palmitate you make through de novo lipogenesis, you will consume 14 NADPH. Those NADPH, which is the redox part of the redox power of reductive power of the cell, is produced through the pentose phosphate pathway. So what we did to demonstrate that fatty acid synthase consume it and that these processes are altered in alternatively activated macrophages is we replace the glucose in our tissue culture medium by deuterated glucose, in which we changed the glucose in position carbon free here of the glucose by a deuterium molecule. That deuterium within the pentose phosphate pathway, the oxidative arm of the pentose phosphate pathway, will be passed to the NADPH, making it 1 Dalton heavier. And when this NADPH is used for lipogenesis, the palmitate produced will be 1 Dalton heavier, 1 or more Dalton heavier, depending on how many of those NADPH has been incorporated into the newly synthesized molecule of palmitate. So using mass spectrometry analysis, we could determine the consumption rate of NADPH by fatty acid synthase in our macrophages. And this is what we've done here. And what you could see here is that the wild-type cell, in response to IL-4, in line with the increased de novo lipogenetic rate, consume more NADPH, which was somehow expected. And if you block de novo lipogenesis here using another fatty acid synthase inhibitors, you decrease substantially the consumptions of NADPH. Eventually, that leads, when you cannot activate SREB1 into your macrophages, to an abnormally increased NADPH to NADP+ ratio. And this increased reductive power in your cell is translated to an increased level of antioxidant deficits of your cells. As you can see here, the SCAP knockout cells have an increased level of glutathione. And when you treat cells with C75, you have an increase of glutathione. So basically, alternatively activated macrophages do more de novo lipogenesis, consume more NADPH, that depletes the antioxidant defensive of the cells. You can see here they're reduced, and that is potentially promoting the amount of ROS. When you cannot do de novo lipogenesis, notably in SCAP knockout cells, you have an increased antioxidant defenses. And we tested that by challenging the cells with hydrogen peroxide. And when you challenge SCAP knockouts macrophages with hydrogen peroxide, what is happening is that they seem to be protected against cell death, such proving that those cells have more capacity to buffer oxidative stress. So de novo lipogenesis in response to IL-4 depletes the antioxidant defenses of the cells. And indeed, this is what we observed when assessing ROS levels into our cells. You can see that interleukin-4 increased the level of ROS. And when you are SCAP knockout, these levels are lower. And we do observe exactly the same when we block fatty acid synthesis pharmacologically with C75. So to finally prove that this was our key mechanisms linking SRABP1 activated de novo lipogenesis with alternative activations, what we did is we supplemented our cell with an acetylcysteine. And acetylcysteine is the precursor of glutathione, and therefore we boost artificially the antioxidant defensive of the cells. Indeed, as you can see here, as observed before, interleukin-4 increase ROS levels in the cells, and in the wild-type cells, an acetylcysteine supplementations blunt these upregulations. However, in SCAP knockout cells that have less ROS to start with, an acetylcysteine supplementation doesn't seem to do much to the ROS level. Similarly, wild-type macrophages treated with interleukin-4 polarized into M2 state, and when you put an acetylcysteine, you reduce the alternative activation of the cells, so showing that ROS are necessary for the alternative activation of the cells, while in SCAP knockout cells, addition of an acetylcysteine is stopping up antioxidant defenses that are already somewhere close to the maximum, and therefore have very, very little effect on the alternative polarization. So it's showing that DNL-derived ROS are responsible for the alternative activation of the macrophages. So just to summarize the first part of my talk, it seems that the process of making fat matters more than the fat produced itself. So what we've shown is that interleukin-4 can activate SRBP1s to trigger fatty acid synthesis in those alternatively activated macrophages. That will produce lipids, but this is not the signal to trigger alternative activation. What we show is that this will consume NADPH until making a competition against the antioxidants defensive of the cells, rising ROS levels to a signaling level of ROS that will permit alternative activation of the cell. So I'm now going to move to the next part of my talk regarding the fatty acid uptake. And yeah, as you remember, we've seen that saturated fatty acids could be deleterious and are associated with the development of cardiometabolic complication of obesity. And we know that the quality of fat matters, but we were thinking of the way what you do with it might matters as well. And what we know from previous studies and a lot of literature is that our cells have a capacity to do something, change the quality of the fat that is inside the cells. So this is named the lipid remodeling, and basically you could transform those saturated fatty acids, such as palmitic acid or stearic acid, that are associated with complication of obesity into unsaturated lipids that are known to be protective. So the question we ask ourselves is, how is that lipid remodeling in adipose tissue macrophages in obesity? And coming back to our models of metabolically healthy obese or unhealthy or obese mouse, that both of them have lipid-associated macrophages, but those one aren't inflamed and those one are. it seems that one of the key pathways that is differently expressed in between those cells is the lipid remodeling capacity. So the elongases and the desaturases that are able to take palmitic acids, take stearic acid and transform it into palmitoleic and oleic acid that we know are protective, suggesting a potential role of lipid remodeling in the development of inflammation in adipose tissue macrophages in obesity. So to test for this, we use an in vitro model, which are bone marrow-derived macrophages as before, and we treated them with stably labeled 13C palmitic acids at rising doses, with those questions in mind. Do the macrophages show an adaptive response to palmitic acids? Can they accommodate lipotoxic in cells? How much those cells can convert, detoxify this exogenous palmitic acid, this lipotoxic palmitate? And is there a link between conversion capacity and lipotoxicity as ER stress or inflammation? So the first things I would like to show you, and it's quite interesting, that if you put lipids, and notably palmitic acids, onto your macrophages, they will tend to resemble the adipose tissue macrophages that is of obese individuals that is also observed in NAFLD livers, as I showed to you during the introduction. So it seems that treating the cell with palmitic acids makes the cell somehow a model that resemble adipose tissue macrophages and NAFLD macrophages, like lipid-associated macrophages. And looking into the lipid remodeling capacity of the cells, what we observe is that the more we put palmitic acid onto those wild-type cells, the more the cells will increase the capacity to desaturate it. They will increase the expression of STD2, steroid desaturase, which is the desaturase of palmitic acids. And because we used in our cell system stably labeled palmitic acid, we could trace the fate of the palmitic acid. So here you can see that the percentage amongst all labeled lipids that has been converted into desaturated lipid is maintained until 50 micromolar of palmitic acid in the cell, at the expense of increasing your capacity to do it at the gene expression level, as you can see here. However, when you reach the plateau in which you cannot increase further your capacity to detoxify lipids, your capacity to actually do it falls. As you can see here, at 100 micromolar, those macrophages are not able to process the loads of palmitic acid that we give to them. We are over competing the product with the substrate. And actually, somewhere in between 50 and 100 micromolar, something is happening. You are overwhelming the system, and your macrophages tends to show sign of metabolic inflammations. You have an increase, this is RNA sequencing data. We have an increased inflammatory activation, as you can see here, increased ER stress, increased hypoxia, pseudo-hypoxic signalings, and increased oxidative stress, as you can see here. So the question we were asking to us, we've seen that in obesity, the adipose tissue macrophages have a lower expression level of the lipid remodeling pathway. So what would happen in these conditions? So what we did is we used again our SCAP knockout macrophages because they tend to resemble the macrophages from the obese adipose tissue. You can see they have strong reductions, notably into SCD1 and SCD2 expression. And we challenge them with palmitate. But first of all, quite interestingly, on the basal states, the fact of not having the capacity to make and remodel fats on the basal stage doesn't seem to impair much the functions and the lipidome of the macrophages. As you can see here, in blue is the wild type, and in light orange is the SCAP knockout, so the cell that cannot do lipid remodeling. You have slight but not quite significant reductions in the level of palmitic acids, and the rest of the lipidome is roughly stable, this being due to reductions in de novo lipogenesis. And I kind of like this slide because this shows you that basically nature is very well made, that basically the Srebs are the regulators of the equilibriums of the fatty acid composition of the cell. They are the guardians of it. And indeed, things are paired. Srebs are pairing de novo lipogenesis and the fatty acid uptake with the remodeling capacity. However, what would happen in the condition of a lipid overload? What would happen in a very lipid-rich adipose tissue? What would happen if we give a lot of saturated fatty acid to the cells? And this is what we've done here. So as I showed you before, this is the wild type cells, and in response to palmitic acid, they increase the capacity to desaturate by increasing the level of SCD2 to maintain the capacity, the ratio of unsaturated to saturated exogenous fatty acids. However, the SCAP knockout aren't able to do it at all. As you can see here, the level of SCD2 start low and are not able to be induced. And indeed, the capacity to desaturate exogenous palmitic acid is low at low dose of fatty acid and stays very, very low. And if the wild type cells were able to defend the lipidome for a period of times until 50 micromolar, as soon as you hit those cells that cannot detoxify exogenous saturated fatty acids, they show all the pathway associated with lipotoxicity, such as inflammations, ER stress, hypoxia, or oxidative stress. In an attempt to try to understand what was the importance of the lipid remodeling into this, we decided to plot the capacity to convert palmitic acid into something else as a percentage of unconverted palmitic acids against some markers, such as CHOP as an ER stress marker. And what we observed is that obviously the more you put palmitic acid, the more you overwhelm the capacity of your cell to deal with saturated fatty acid, and the more you accumulate the palmitic acid that you put on the cell stays at such, and the more you increase ER stress. However, by putting as a covariate the percentage of unconverted palmitic acids, we are normalizing somehow the wild type and the knockout. So all the differences that we were observing between the wild type and the knockout root to the capacity of the cells to process exogenous palmitic acid and transform it into something else. And this is true for ER stress marker, but also to the loss of the anti-inflammatory profile of the macrophages, as you can see here. So basically, the capacity to convert palmitic acid into other lipids correlate with ER stress and M2 polarizations, independently of the dose applied to the cells, because the key variable here is not how much you put on it, but how much you can deal with it. Basically, the metabolic fitness of your cells to accommodate saturated fatty acids load. So to go slightly further into the characterizations of this lipotoxicity and the role of the fatty acid remodeling, we use the differential expression data of the wild type and SCAP knockout macrophages in response to lipotoxic incest to palmitic acids. And from those cells, from the same very same samples, we measured the desaturation capacity of the cells. And we used this as a covariate to determine which pathway were actually strongly dependent from the capacity of the cells to do fatty acid remodeling. And actually, we corrected the vast majority of the differences by using the remodeling capacity as a covariate in our analysis. And we perform RNA sequencing analysis on the genes that has been modified. And this is the data that we found. And this is we found some very expected one. And as we see, like the ER stress response is normalized, the inflammation is normalized, the ROS is normalized, but quite interestingly, the key pathways that were normalized were not the expected one. It seems that fatty acid remodeling is very important for cholesterol homeostasis, but also from autophagy pathway with very strong activation of mTORC1, and as example, but also cell cycle signaling with p53 or beta-catenin. pathways. So we are now investigating the role of those pathways in mediating the fatty acids remodeling and also in mediating the defects associated with impaired metabolic, impaired lipid remodeling that is observed in obesity. So if we can't fix the capacity of the cells to perform, to detoxify the lipids, can we fix the consequences of it? That is basically the pathway that I'm showing to you here. So just to conclude quickly, we show that in murine model of obesity, adipose tissue macrophages have biphasic expressions of the fatty acid remodeling program, which is upregulated in healthy obesity and downregulated in unhealthy obesity. We've seen that macrophages can enhance the desaturation capacity when exposed to lipotoxic insults, and that impairments of lipid remodeling render macrophages extremely sensitive to lipotoxicity, and that fatty acid remodeling can predict the lipotoxic response. And somehow, the key take-home message I'd like to give to you from this part is that it doesn't matter how much saturated fat you eat, as long as you can deal with it. And this is what we are doing at the moment in the lab. We are trying to obtain blood monocyte and monocyte-derived macrophages from different people with different stage of disease, with different stage of obesity. We challenge them with this labeled palmitic acid, and we assess the lipid remodeling and the gene expression, trying to see if we can assess metabolic fitness of the macrophages and if that metabolic fitness is associated with the occurrence of cardiometabolic complication of obesity. So I like to thank the people who participate in those studies. So in particular, Tony and Sam, but also Martin, Renata, I mean, all the people that have been helping. Our collaborators in Heidelberg University, Julio and Aurélien, who are responsible for the bioinformatic analysis, collaborators at LMB and Trinity College Dublin for the Helminth work and our funders. And thank you very much for your attention, and I'm very happy to take any question. We are trying to access as much as we can to adipose tissue macrophages data from obese individuals and trying to assess the expression of it. One of the things that we do observe in the preliminary results that we've done with human cells ex vivo, but they are blood-derived cells because obviously it's not always easy to access to fresh adipose tissue macrophages, is that the expressions of SCD do not necessarily correlate very well with the activity of the enzyme. So basically, it seems that sometimes actually the more you try to increase it is because your capacity to desaturate your lipids to start with was bad. Mm-hmm. So basically, people with higher expressions, like not in a genetic model like we use in SCAP knockout, in which it's completely downregulated, people will have these moments in which they will increase the expression, but still not being able to deal with it. So ideally, what we would like to do is we are using blood-derived monocyte and macrophages, eventually moving to people with obesity or with diabetes or with fatty liver disease, trying to see if we can measure their metabolic fitness. And maybe eventually, if we have access to liver macrophages or adipose tissue macrophages, we can test that as well. But obviously, it's very precious and rare material, and we are not having a link with clinic. In the adipocyte, that's a very, very good point, and I agree that de novo lipogenesis is decreased in obesity in the adipocyte. I don't know about the ROS, and the thing is measuring de novo lipogenesis in the adipocyte is very difficult somehow because all your signal is diluted in the amount of fat. So if you use deuterated water or to measure it, you have the issue that your deuterium is diluted in the amount of fat. However, we are investigating somehow. We are trying to see if we could see that our mechanism is valuable in the liver cells, that if you have an increased de novo lipogenesis in the hepatocyte, does that lead to more ROS? And if that could lead to the development actually to the transition from NAFLD to NASH, that we know is associated with an increased ROS levels in the liver. So this is we have very preliminary results at the moment. There were a student working on it. It's promising, but very preliminary at the moment. Right. What we think is that autophagy might be beneficial because that might be able to alleviate some of the lipid load and potentially get rid of the because what we observed that I didn't mention is that when you put the palmitic acid, cells starts to do some fatty acid oxidation. And it seems that on the long term, those mitochondria may become dysfunctional, also because they will start to accumulate those saturated fat in their mitochondrions. So the idea is that if you increase autophagy, you will allow the systems to keep running in a better way by discarding the bad mitochondria, if I can say. We are currently doing some experiments using rapamycin, and it seems it's a very difficult one because when you use rapamycin, you don't activate the desaturation system. So one of the key experiments that we would like to do is trying to see, does rapamycin can rescue those CAP knockout cells? And this is, I don't know yet. That's a good question. And for many, many years, we are trying to compare those two. And it seems they're actually very, very, very different kind of cells. That's the fact of accumulating a vast amount of cholesterol will have a lot to do with SRBP2 and LXR activations, while the triglycerides component is very much PIPA gamma SRBP1 pathways mediated. So unfortunately, there is very, very few overlap in between the two, but that makes it very interesting because if it was exactly the same as in the atheroma, the field will have died very, very quickly, I'm afraid. Okay, the ER stress have two effects in the macrophages. What we have seen is that a relatively low level of ER stress is actually beneficial because that is activating the SREBs. We believe that actually it's one of the key signals to activate the SREB pathway and allow the macrophages to desaturate the lipids. That is actually part of the whole sensing machinery, that your lipids will get uptake, your saturated fatty acids be incorporated into membrane lipids. That will basically the general models change the curvatures of the ER membranes, activate ER stress to activate the SREBs and allow the systems to accommodate. If that replies to the first one. And obviously on chronicity, which is what's happening in obesity, this ER stress tends to activate junk, so therefore inflammations, and eventually ER stress-induced cell death. And it's known that macrophages cell death in obesity is associated with worsening of adipocytes and adipose tissue function, if that answers the second part of the question. Okay, what we tested is the origin of the ROS inside the system. Okay. So basically, we compare classically activated macrophages with LPS with alternatively activated macrophages. The alternatively activated macrophages seems to produce their ROS from the mitochondrion because the mitochondrion are more active in alternatively activated macrophages. This has been published by several papers. And when we measure mitochondrial ROS, they are upregulated. We do not observe any changes in NADPH oxidase ROS, xanthine oxidase ROS, and so on. By using inhibitors, we could not modulate those levels. What we think is happening, therefore, is that ROS are produced in the mitochondria, but your cytosolic buffering capacity is lower, so therefore they tend to accumulate in the cytosol. The way we believe it works is like you allow tiny amounts of ROS, and one of the potential mechanisms it will ask on phosphatases. And what we did observe, but it's not in the manuscripts, but other people have been showing similar results in other kind of cells, is that will allow more sustained activation of STAT6, which is one of the key transcription factors involved for alternative activations, by removing the capacity of the cells to dephosphorylate it. Okay. So basically, and this is how the ROS will signal. Okay, that's actually a good question, because there is currently clinical trials targeting SCD, trying for the treatment of NAFLD. We actually use this Amarcol, if I'm correct.
Yeah.
And we did some preliminary experiments using Amarcol onto human macrophages, actually. And we do observe that when you do this in the context of a lipid challenge, those macrophages become very inflamed. You have interleukin-1 beta that is very high. And I think if this data would not necessarily guide the fact of like, we need to enhance SCD activity or whatever in the macrophages, that can also inform to pay attention when we target those pathways in other cells, that the macrophages composing the liver will be exposed as much as the hepatocyte, and therefore will become more inflamed. And I think that's always very important. That's we consider always all the other cells. To go as well to the first part of your questions, what we're looking at the moment is, first of all, macrophages have very active lipid metabolism pathway with very few latency, which is what is the problem when you work with adipocytes, as example. There's a lot, because there's a lot of lipids, it's very difficult to assess fluxes. That if you could try to identify the people that are more susceptible to develop inflammations or ER stress or whatever in response to palmitic acid, we may be able to identify the people that are more susceptible to go toward NASH, to go toward type 2 diabetes, having like subpopulation that should be looked at more closely. And because we know that there's a lot of obese people that will never develop complications, and there's a lot of obese people that will have very strong complications at a lower BMI than the rest. And we don't have a good way to know those people. So at the moment, we are actually seeking for a biomarker potentially. But yeah, it's quite difficult, because at the moment we need quite a lot of cells to do this lipid tracing assay, and therefore quite a lot of blood, which is not always available. Yes, potentially. One of the things that we know, and I didn't show this data, is that inflammation reduces very potently your capacity to deal with lipids. So what we observe is that if we pre-treat your macrophages with LPS, it's somehow a wrong challenge, because it's like a bacterial infection challenge. You decrease the capacity of the cells to process lipids, but like to zero, they become extremely sensitive to the lipids. So one of the key of the way that I would think is to train will be to target inflammation. Because if you target inflammation, you will render your macrophages less sensitive to palmitic acids-induced lipotoxicity, and therefore make your cells fitter to deal with the lipotoxic load.
Guillaume, thank you so much again for sharing your expertise today. And thank you for being here. And once again, I would like to thank the organizers and the sponsors, and for sure all of you for attending, for contributing to the discussion. And I hope to see you all in the next seminar. So goodbye and take care, everyone.
Goodbye. Thank you very much.
Bye.